Bueno, entonces ya nada más, como ven, ahí aparece en la parte de arriba izquierda que se está grabando en YouTube. Es un video privado, no va a salir en YouTube ni nada, va a ser para subirlo a la plataforma después. ¿Vale? Entonces, a partir de la siguiente clase, la mayoría de las clases van a estar grabadas para que quienes tengan la necesidad de seguirlo solo en plataforma y no puedan conectarse, puedan tener acceso a ese material. Okay, so I'm going to shift to English now. So for today's class, I will need you guys to participate a little bit more. So I will be asking questions. I would like you to speak out, to say what you think. So I'm gonna share my slide, but I would like you guys to help me with this class because I need to know how much you remember, how much do you know, what do you think about the gene therapy, and a few things that we're going to be talking about this in this session, okay? So in the previous class, we were talking about very quickly and just not very well organized manner about DNA structure, genes, chromosomes, chromatin. So we're going to go back to some of those topics today. So the first question I would like to ask you guys is, what do you think, what gene therapy is? So anyone who wants to tell what you know, or what's the concept you have about gene therapy? Okay, let me see your names here. Ramon, what do you think about gene therapy? If you're going to speak, please turn your cameras on so that I can see you are there. Otherwise, I'll be just speaking and maybe you are not there. So anyone who wants to tell us what you know about gene therapy? Regina? Carlos?
What is gene therapy?
Yeah. I don't need a formal definition. I just want to know what you think gene therapy is. What do you understand by gene therapy? So I want to hear what do you think about it? Carlos, I think you wanted to say something.
Well, gene therapy is a whole bunch of techniques that aims to modify cells outside the body. Using gene transference, for example, no?
Right. Wenceslao?
I think that it's a mechanism to improve the health of many people through novel mechanisms.
Okay. Anyone else? Petra?
I think are techniques for correct genetic disease.
Okay, genetic diseases. Jose Fernando?
Maybe it's the modification of DNA, DNA that occur disease.
Okay, modification of the DNA. Wenceslao, you have another something else to say?
No.
Okay, Regina?
The gene therapy is the mechanism in altering the genes inside the body in to treat or stop disease.
Okay, so if we just think about the name gene therapy, it's a therapy for a gene or something like that. So therapy means we're going to treat a disease or a disorder. So we want to cure or to improve the function of the body, of a cell or something. And it's focused on genes. So it's different to other therapies because here the target of the therapy are our genes. Okay, so I'm just going to show you here a definition about gene therapy. So it's not the only definition, and it's not most probably complete definition, but it's a technique that modifies the genes of the person's genes to treat or cure a disease. Okay, so as you guys were saying, so we are talking about the techniques, or as Carlos was saying, it's not only one technique, but it's a collection or a bunch of techniques that are used to modify our genes. And not just because we want to modify the genes, but because we want to cure a disease. As we will see in another lecture later, there's another approach of using gene therapy that is not to cure a disease, but to enhance capabilities of the body. So I don't know if you have read or if you heard something in a movie or in the news that in some countries people think that countries like China, for example, are using gene therapy to make their soldiers stronger, faster. So we're going to talk about that in another session. But actually, the idea is that gene therapy, it's a collection of techniques that we use to modify the genes of an organism. In this case, we're talking about human diseases or human conditions. Okay, and there are three main goals that we would like to reach with these techniques. One is to replace a disease-causing gene with a healthy one. So if there is a gene that is malfunctioning, that is functioning, its function is altered, and that altered gene is giving rise to a disease, we would like to change that disease-causing gene with a healthy one, with a normal gene that is not going to be able to cause or to favor the disease. We will also like to inactivate a gene. So one is we can exchange the disease-causing gene by a new one that is healthy, or we can inactivate that gene. We are not going to exchange it by a new one, but we just want to inactivate that. Or we may want to introduce a modified gene into the bodies to help treat disease. So these are different approaches that we can use. So we can actually exchange the gene, the altered gene, for a healthy one. If the gene is altered, maybe we don't need to replace it by a healthy one. We just inactivate that. Or maybe there's a disease, and we can introduce a new gene or a modified gene that will help our body to fight that disease. So these are like three major goals that we try to go to these three things when we talk about gene therapy. So if gene therapy is a technique or techniques to modify genes, so then what is a gene? How do we define the gene? So who wants to give me a definition of a gene? Come on, guys, I want to see many hands up. That's basic molecular biology. How do we define a gene? Angel, David, how will you define a gene? Wenceslao?
It's a coding region of the DNA.
A coding region of the DNA. Okay, it's an idea. Carlos, you wanted to?
No, it's a sequence of DNA located in a locus on a chromosome.
Okay, so now we are talking about a gene. It's a region or a sequence of a chromosome.
That's right.
It contains information. So
yeah, indeed, a gene codes for proteins.
For many years, when we were talking about genes, yeah. So the definition of a gene for many years was just a region of the genome or a region of a chromosome or a DNA region that contains the information to produce proteins. Now, today we know that there are regions in our DNA that do not necessarily contain information to produce proteins, but they just have information to produce what we call the non-coding RNAs. Okay, so we have the genes, in the DNA. We're seeing another slide a little bit, it's more complex than just a fragment of DNA, but let's think about the fragment of DNA, which the sequence in that DNA contains a code or the information to produce either a messenger RNA, which will be later used at the ribosome to produce a protein, or it can be just a non-coding RNA. So these RNAs are molecules that will never go into the ribosome to be used as a template for protein synthesis, but they are very important because they play regulatory roles in cell functions. So we can actually define the gene, like the genes are part of our genome. These genes encode the information for making proteins or RNAs. Is what we were just saying. So it's not only sequences that encode for proteins, but it's also there are genes that are that contain the information for RNA synthesis. That is not messenger RNA, non-coding RNAs. Okay, so then if the genes are part of our genome, so the next question is, what is a genome? So what do you think a genome is? What's the definition of the genome? You know, there's this huge from humans to understand what are the sequences, the codes, the genes. So how we do define the genome? Jitsar?
Yeah. The definition that I would give it would be like the whole information, the whole genetic information of a certain species. Yeah, would be like kind of like that.
Okay, the whole genetic information of a given species, or Carlos? Oh,
Is the whole information from an individual.
okay.
The genetic information, of course.
Right. So if we go to for a definition, it's the genome is the complete set of genetic information in an organism. That's basically the definition of the genome. So when we talk about the human genome, it means all the genetic information that is in our cells. The mouse genome, the plant genome, the bacterial genome, it's all the DNA, all the information that is contained in the DNA that is required for the organism to function properly, okay? So, and then you see, it provides all of the information the organism requires to function. In living organism, the genome is stored in chromosomes. So we have another definition to go for. So now, what are chromosomes? So we know, I'm going to go back a little bit to the slide. So we started with what is gene therapy. We were saying that it's a collection of techniques, genes. And then we said, okay, what are genes? So genes are parts of the genome that encode for the formation of proteins or RNA molecules. And what's the genome? So genes are part of the genome. The genome is all the collection of genetic information in our cells, and that's packed or stored in chromosomes. So what are chromosomes? So chromosomes are single long molecules of DNA. So basically, our genetic information, like as humans, in previous class, I think we were saying that we have 23 pairs of chromosomes. So each chromosome has a lot of genes, and those genes contain the genetic information that is required for our body to function properly, okay? So when we talk about the chromosome, chromosomes are long DNA molecules. And we were saying, I think in the previous class, that if we were talking about prokaryotic cells in bacteria, the chromosomes are circular DNA molecules, but in eukaryotes, these molecules are linear. So we have a linear molecule, very long molecule, it's a chromosome. And that contains the genetic information. And that genetic information, if we go back to the definitions that we have here, it's our genome. So our genome, it's all the information that is in our cells. It's organized in chromosomes. And the chromosomes are like the basic unit of storage for genetic information, okay? So why am I going through all these basic definitions? Because first slide here, so it's important to understand what genes are, because gene therapy, it's the goal of gene therapy, it's to modify genes. So we have to know what genes are, how are they organized, what's the composition of a gene, how are they organized in a chromosome, what is a chromosome, and how all this collection of genes that are stored, organized in chromosomes, constitute our genome, okay? So I'm gonna go further here. So this is a nice picture of how our chromosomes are organized. So we have DNA, double-stranded DNA. As we were saying in the previous class, the DNA is packed in this very nice structure. Do you remember how are these structures named? So there are histones in the middle and DNA around. How are these called? Mrs. Lau?
Nucleosomes.
Okay, these are the nucleosomes, and they are all the chromosomes have nucleosomes all along the molecule of DNA, and then they can have what we call the supercoiling structure, where these are going to be packed and coiled around each other. And during mitosis, we have these very nice sister chromatids that are arranged in an X shape. That are the ones that are going to be segregated to the daughter cells. So if we think about, okay, I want to modify a gene, because this gene, if we go back to this slide here, says, okay, I want to replace a gene that is causing a disease, or I want to inactivate the gene. So how can I go and replace that gene? How can I inactivate that gene? If we go back to that slide that I was showing, in this single chromosome, there will be hundreds of genes. So how do I know where that particular gene is? How do I know, how can I exchange that gene? How can I inactivate that gene? So to finish up with this chromosome story, I just want to point out that the chromosomes in eukaryotic cells have three major components that we already mentioned last class. It's the telomere, replication origin, and centromere. And they're important because, as you will see later, and this is actually something that I want to ask you. So centromere, replication origins, and telomeres are sequences, are structures that have specific sequences. So what do you think will happen if in a chromosome there is let's say that our cells get exposed to X-ray or UV radiation or whatever, and the sequence that it's here at the centromere, it's mutated. So those sequences that are at the centromere, suddenly, because our cells were exposed to either a physical factor like radiation or chemical factors, the sequences there are mutated. They change sequence, or there is a deletion, or there is an insertion. What do you think will happen to the chromosomes if those sequences are altered? Anyone has an idea what may happen if we change the sequence at the centromere? No, you don't imagine what will happen if we mutate the sequence at the centromere region, or what will happen if we mutate the replication origins or the telomere? So for that, you should know what is the function of the centromere. So as you can see in these pictures, when this first picture is a double-stranded DNA, that's a chromosome. During these phases here, the chromosome will be replicated, So we have now two copies, identical copies of the same chromosome, and they stay attached by these structures. In these pictures, we don't have the full picture, I would say, but here they are not attached just by DNA to DNA. So there are lots of different proteins that are involved in that attachment. So here there are protein complexes that will bind to the centromere, to the other centromere, and they will keep the centromeres together. So what will happen if we mutate those sequences? So,
Well,
yeah, Carlos?
in the in the context of DNA condensation or DNA recombination, is a kind of a handbrake for the mitotic process. If you like to stop somatic cell that you find some serious markers, sorry, perhaps you are doing research, basic research, and you want to know which kind of substance works better in order to stop the mitotic process. I don't know. Perhaps it's an idea.
Okay, yeah. So, yeah. Yeah, actually, my question was thinking about if our cells are exposed to something that changes the sequence here, then all the proteins that usually bind here and that will help the two sister chromatids to stay together, the proteins will not be able to bind anymore, and we may see chromosomes that are detached. The single chromatids are not attached. And that will have a lot of problems. The cell will have problems with their mitosis because the chromosomes are not going to be segregated properly. So that's an example of what may happen if we mutate those regions. And for the replication origin, it's probably easier to imagine. So if you remember, replication origins are also sequences that are recognized by proteins, so that the DNA can be separated, the two strands, and then the replication machinery can get there and start replication the DNA. So what may happen if we mutate the DNA replication origins? So those proteins may not be able to bind, and probably the cell is not going to be able to recognize where the replication will start. And maybe we don't have DNA replication. And then we are in big trouble because when cells divide, they have to replicate the DNA. So this is just a few examples of what may happen if we mutate certain particular regions of the chromosomes, okay? So let's move on. So now the thing is, we are talking about chromosomes, genes, gene therapy, and we mentioned that gene therapy, it's a technique that by which we want to modify genes, but genes are fragments of DNA. So, and what is DNA? So DNA, more specifically, it's a deoxyribonucleic acid. And do you guys remember why it's a deoxyribonucleic acid? Why is an acid? And why is a deoxyribonucleic acid? From this structure here, what gives the molecule the acidity and why it is called ribonucleic, deoxyribonucleic acid? Anyone? So as you remember, there are three major components of nucleotide. Nucleotides are the monomers that will form the polymeric molecule that is the DNA. So we have the core molecule, the sugar, it's in the middle. We have phosphate group on the left-hand side, and we have the nitrogenous base on the right-hand side. So these are the three major components. So the acidity of the molecule comes from the phosphate group, okay? The phosphate group gives the acidity to the molecule. And we call it deoxyribonucleic acid because this sugar here how is this sugar called? You remember, guys? What's the name of this sugar?
Ribose.
In this case, it's a deoxyribose. Why? Because the ribose will have an oxygen here. It doesn't. RNA molecules have ribose with an oxygen here. This is deoxy because that oxygen is not present there. So we have only oxygen here in the carbon at the position number 3. Okay, so that's why it's called deoxyribonucleic acid. So this is the basic unit of the DNA. And so as we were talking in the previous class, so this is just a brief reminder of how DNA is organized. So there are a few key features that we have to keep in mind. So first, it's a nucleic acid. Specifically, it's a deoxyribonucleic acid. Then it's a polymeric molecule. Why polymeric? Because the monomers, which are the nucleotides, are bound to each other in long, very long chains. So these are polymeric chains. And we have a molecule that is double-stranded. So we have one strand here on the left and a second strand on the right. And both strands are very similar because the backbone is formed by covalent links between the sugars. This sugar here is bound by a phosphodiester bond to this sugar there, this one to a phosphodiester bond to the next one, and we go on like that. And the same happens on the other strand on the other side. So there are two strands. That's why it's called double-stranded. The backbone of each strand, it's the sugar backbone, and in the middle, in the inside part of the molecule, we have the bases, which are forming hydrogen bonds. So that's basically the basic structure of the molecule. And why is this structure important? So as you know, the sequence, how the nucleotides are organized, like here, A, C, T, C, A, C, C, that's the code. The order, how the nucleotides are organized, defines a message. It has a message. If we change the message, we may change the function of the cell. And if we go back to this slide, so we mentioned again, techniques to modify a person's gene to cure a disease. So we have to know where the genes are and what's wrong with the gene. So we need to know what's wrong with the gene so that we can repair the gene if we need to repair the gene. And so what's the relation between genes and disease? So there are many diseases that are that have a very important genetic component. So these are basically diseases that can be transmitted from one generation to another one, but there are also diseases that are not inherited. So we can acquire a genetic transformation or a genetic defect that gives rise to a disease, and that's it. It doesn't come from our parents. It may happen, probably it happened during embryonic development or in adult life. So to understand how genes are related to disease, we have to remember about the central dogma of molecular biology. Okay, so I asked you to read a little bit to remember the central dogma of molecular biology, and I would like you guys to tell me what do you remember of if you read something about the central dogma of molecular biology.
Well, in short, DNA transcripts to RNA, and translate to protein, or DNA to RNA to protein.
Okay, so I'm just looking for the. So I think you guys, if you are familiar with the Zoom application, on the top of your screen, you will have a button where you can annotate. If you click that, you can write text or draw things in the screen if you want. So basically, we have DNA, DNA will be transcribed to RNA. Is that correct? And RNA will be used as template for the synthesis of proteins. Okay? It's a little bit difficult to write with the mouse, protein. Okay? So what else do we have to add to this picture to complete the central dogma of molecular biology?
A replication of DNA.
Okay, something very important, the DNA, it's able to self-replicate. So there are mechanisms in the cell where the DNA can make an identical copy of the previous molecule that is there. So DNA can make other DNA molecules, then DNA can be used as a template for the synthesis of RNA, that is called mRNA, the messenger RNA, and that messenger RNA will be used, will be taken to the ribosome, where it will be used as a template for translation. That is the process where RNA is read into protein synthesis. And as we were, so this was like the original idea of the central dogma of molecular biology when it was described. But now we know that DNA can give rise to RNAs that are not going to be used for protein synthesis. So what we call the non-coding RNAs. So there are non-coding RNAs. So that's something different from the original dogma that was described. The original dogma was DNA makes DNA, DNA is transcribed to RNA, RNA goes to protein. Now we know that there's RNA that will never go into protein. And now there's something else. Is there, what else can we add here that we know now that may happen with these molecules? So now we know that from RNA, we can go back to DNA. Anyone knows how this process is called? You know what's the name of the process where DNA is synthesized from an RNA template?
Reverse transcription.
Reverse transcription. So what are the names of these processes that we have here? So we have, I'm going to write here, replication. A DNA is used as a template to make a copy, identical copy of another DNA. So we have transcription, which is the synthesis of an RNA molecule from a DNA molecule as a template. And then we have reverse here, reverse transcription. Reverse transcription. Okay, we have reverse transcription, and then we have down here translation. So those are like the four basic mechanisms that are involved in the central dogma of molecular biology. Okay, so as I was saying, the original idea of the dogma was this one here. So DNA, transcription to RNA, translation to protein, and then we were just saying now that we can have a very important process that is DNA forms another DNA molecule. And now we know that the DNA to RNA process is reversible, so we can go from RNA to DNA. Okay. So why is this dogma or this process important in relation to genes and disease? So why do you think understanding the central dogma is important to understand how genes are related to disease, or how diseases are originated by alterations in gene structure? So what will happen if there is a mutation that eliminates this base pair here. So we may not have these nucleotides here, and we may not have this protein, this amino acid here. So that will give rise to an abnormal protein. Okay, if we have abnormal proteins, the cells are not going to function properly, and that misfunction of the cells may originate a disease or contribute to the development of a disease. So,
well, I
that's
think
why gene therapy. Yes, Carlos.
sorry. I think the process of base tautomerism is a kind of cause of those modifications in the nucleotides. The nucleotides, sorry.
Yeah, that's one of the processes that can give rise to alterations in the DNA sequences. So I'm going to go to the next slide because I think I have there some examples. So this slide is just to tell you that, okay, why understanding this is important, because whatever happens at the DNA level will have a consequence at the RNA level and at the protein level in case of genes that are protein coding genes. And for those genes that code for RNAs, it's the same. So whatever happens at the DNA will affect RNA. I just want you to think a little bit. Just imagine that this process is happening in the cell, and then our cell is exposed to some chemicals or whatever, but those chemicals are something that will destroy the RNA. So if the RNA is destroyed, we won't have any protein, and we have a problem because our cell now is malfunctioning. But we still have the normal DNA. So normal DNA can produce more RNA, and where those factors, toxins, chemicals, or whatever are gone, if our body is clean again, our DNA is still normal, and it's going to be producing new RNA that's going to be perfectly fine in proteins, okay? So if something affects the RNA or the protein, it has an effect, but it doesn't matter so much because we still have our genes that are going to be producing more. The problem is when those alterations, point mutations, deletions, whatever, happen at the DNA level. Why? Because those alterations in the DNA, when our cells will divide, they will pass the alteration to the next generation of cells. And if that goes into the germ cells, we will pass that to our daughters and sons. So that's why DNA is so important. So if something was wrong with the RNA, it's no big problem, but if something is wrong with the DNA, it's a huge problem because it's something permanent that's going to be there forever, and everything that comes from that gene, every RNA that comes from that gene is going to be altered, okay? Okay, so let's move on with the next slide. So I'm just going to go, these slides, I'm going to go quickly just to remind you the complexity of the DNA. So last class and today we will be talking about a chromosome, that is a long DNA sequence that contains the genetic information. Those are packed in around histones, and they can have very complex organization like in the mitotic chromosomes. Those chromosomes are going to be segregated from one cell to another one when replication occurs. And something important here is the genome, it's very, very complex. So when we look at these chromosomes, they look very nice, like an X shape, it's very beautiful. And if you have seen some karyotype pictures where they paint the chromosome from different colors, it looks very nice. And we have 23 nice pairs of chromosomes. But if we go deep into that chromosome, we will find that it's very complex. Every single chromosome, if we take this tiny part of the chromosome here, we have all those genes. Every name that you see written here, ABCD1, PXNB3, SRPK3, every one of those, it's a gene. Okay, so see how many genes are there in a very tiny part of the chromosome. Okay, so when we go back here now, you can imagine in every single part of the chromosome, there are genes. And there are many, many different genes in every single chromosome. Do you guys know how many genes approximately we have in our genome, humans?
20,000.
20,000, 25,000. We are still sequencing new regions of the DNA that we are finding new genes. But this is approximately 20, 25,000 genes in our genome. And those are organized in different chromosomes. So just imagine, okay, there is a genetic disease. There's a disease that you just find in a population. And you just realize that that disease is going from one generation to another one. Hey, it's a genetic disease. It's passing from one generation to another. What's the causing gene? How do we look for that gene? How do we know which among these 20,000 genes is causing that disease? It's not an easy task to do, okay? And then if we find that gene and then we want to repair the gene, it's an even harder process, okay? So if we look at a single region of that, okay, we go from this X chromosome here, we take that tiny region of the chromosome there, we have only two single genes here, gene A and gene B. So what happens very often is that even if those genes are in the same chromosome and they are very close to each other, relatively close in the genome sequence, maybe the cell needs a lot of protein A, so the gene is very active, gene E, but gene B is not too active because we don't need a lot of B protein. And that's very dynamic. At some point, maybe gene A is going to be repressed and we will decrease the production of A, and maybe gene B will be increased, activated. So this is something dynamic, okay? So something else. So if we'll, here we have the representation of a gene is just like this yellow orange region, but genes are more complex than that. Genes are not simple things. So if we look at the gene in eukaryotic cells, we have the regulatory region of the genes, and we have the coding sequences that are the exons separated by non-coding sequences that are the introns. And the number of introns and exons and the size of the exons and the introns that are in a gene are very variable, okay? So I'm gonna go, so just before going to the next slide, this is something very important. In our genome, there are very few protein coding genes, and we have most of the DNA sequence in our genes is non-coding DNA, okay? So non-coding DNA for many years was thought to be like trash, it doesn't do anything, but now we know with more technology and investigation, we know that the non-coding function of the cells. So at the end, the gene, it's a discrete or a small linear set of nucleotide pairs. That's a gene. So many nucleotides, one after the other, in a particular sequence, and it has a regulatory region and a coding and non-coding region, intercalated introns and exons. So how can we identify genes that are involved in disease, and how can we exchange or modify those genes? So there are many challenges for that. And before going to the challenges, I'm just going to go quickly to this. So what can go wrong with the genes? So we have the genes, we have the chromosomes. There are many genes in each chromosome. In our cells, they are in the nucleus. They are protected by the nuclear envelope and by histones. So they are very well protected. But even though they are protected, the chromosomes, things can go wrong and chromosomes can get altered. So one natural process where chromosomes get very often altered is during mitosis. As you know, mitosis, we have, first we make a copy of the chromosome, we have to condensate the chromosome, and we have to align the chromosome at the middle of the cell, and then we segregate them to both daughter cells. So very often, not very often, but sometimes during this process, one of the chromosomes is not well attached to the microtubules, and will left behind, and then one cell will have more chromosomes than the other one, and one cell will be missing a part of the chromosome. So as you can imagine, if one cell is missing a chromosome, it's missing maybe 100 or 200 genes, not only one gene. So that's cell is going to have a big, has gained a lot of genes. So that's a very dramatic change that may happen sometimes during mitosis. Something else that can happen not in mitosis, but during meiosis, that is we're going to see that in another class when we talk about diseases that are originated during embryonic development. The chromosomes can recombine. Two chromosomes can get close to each other and they can exchange parts. So now we have new chromosomes with different organization of the genes. Other process that can give rise to alterations in the DNA sequence is DNA replication. As you know, DNA replication is we take one double-strand DNA, it's separated, we have DNA polymerase that binds here and starts moving that and making a copy like you see here. We have the original strands up here. They are separated and then new strands are formed in both sides. Well, now we know that the DNA polymerase can make mistakes, can introduce nucleotides that are not the perfect match to the one that is there, and that can give rise to point mutations. We can have mutations that are caused by exposure to UV radiations, for example. We already mentioned this in the previous class, and we said that there are different types of mutations. Mutations are changes in the DNA sequence, and they can be point mutations, deletions, insertions, and they can be spontaneous or induced. So we already talked about these last class. So I'm going to go to just to remind you these, that the point mutations are changes in one single nucleotide or base pair. Deletions, we eliminate a set of nucleotides, and insertions, we have new nucleotides coming into the sequence, and that can be originated by errors in DNA replication or repair, or by physical or chemical or biological agents. So I'm going to go, okay, I'm just going to go here. There. This is what Carlos was saying. So this tautomerization is a process where by chemical modifications, the structure of these bases can be modified, and then they will chemically they will acquire a different form that when the DNA replication happens, these nucleotide instead of cytosine will be recognized by as another different nucleotide. For example, where is the looking for? Okay, here on the bottom, let me annotate it for you. So this one here that I'm gonna highlight, that's a cytosine. Okay, you see there's a ring here, there's the amino group here, and we have an oxygen here. And if we compare with this one there, what's the difference that you see between those two structures? Just look at them and tell me what's the difference between the two structures that are just surrounded by this pink line.
The position of the position of the carbon atom.
Okay, if you see here, there is a double bond, the nitrogen with the carbon, and here it's only single bound. Okay, that's the first difference, and that results. And as a consequence of that, here in this one, nitrogen here, this nitrogen here, you see this yellow line, it's binding to hydrogen molecules and to the carbon here. That's the cytosine. In this immunoform of the cytosine, what happens is that there is no longer this hydrogen here. So we are losing one bond here, and that bond will move here. And then we end up with the nitrogen that is here. So we still have the nitrogen. I'm going to delete all this. So we have the nitrogen here binding to this hydrogen. They are here. Because we don't have this hydrogen there binding here, this bond will be formed as a double bond here, but it's there. And then this nitrogen here, it doesn't have anything else here, but now it has an hydrogen there. So there are I would say one major change is this hydrogen is no longer here. Now it's here, and then double bond here. That's a very small chemical change, but that's enough to change the properties of binding. So usually cytosine will bind to guanine, but here, because of this change, now it can bind to adenine. So we can, during DNA replication, if there is a tautomerization like this, there's a cytosine, is converted to this immunoform of the cytosine, and then when the DNA polymerase comes, instead of binding the complementary in a change in the sequence of the DNA, in the original sequence, okay? That's a process that happens not very often, but it happens in the cells, okay? And that can give rise to changes in the sequence of the nucleotides in the DNA strand. And similar to this, there are many other, this is an example to that what will happen if we have these tautomers here. We will end up having the original sequence, but then we will have a mutant sequence after 2 rounds of replication. At the first round of replication, this is a normal sequence. On one side, we will have the normal copy, but in the other one, we have the tautomeric thymine binding with the guanine. In the second round, thymine on the top may be recognized by an adenine, but now we have a guanine here that's going to be recognized by the cytosine, and now we have a new base pair here that it wasn't there before. Okay, so that's one of the forms of how we can get mutations in our cells. And there are other more complex and sometimes more dramatic mutations, like we have something that is called 3 nucleotide repeats. So it means sequences of 3 nucleotides that are repeating many times in the sequence of the DNA. So like here, we will have CGG 56 times consecutively. CGG, CGG, CGG 56 times. And that's what is called the wild type allele in this example. But some individuals may have more than 56. They may have up to 200. And some other individuals will have more than 200 copies of that repeat. And in some cases, that having more than 200 copies will result in a disease. And we will see that in one of the next classes where we will talk about genetic diseases, okay? And how those nucleotide repeats expansion happens. So basically, the polymerase goes, reads one copy, another copy, and then because they are complementary, they will form these loops, and they will pull down the string, and then the DNA polymerase will pass again it was already there, but it goes again, making copies of the same region many times. That's another form that we can get mutations or changes in the sequence of the DNA. So the final slides are just what is the biological effect of a mutation in the DNA. So we can have mutations, but so what's the effect of the mutation? So the answer is it depends on many things, many factors. First, what type of mutation it is. So if it's a point mutation, we are changing only one nucleotide. It may not have a dramatic change in the function of the gene. If it's a deletion or an insertion, it will also depend how large the deletion or the insertion is. But it doesn't depend only on if it's a single point mutation or not. It depends what type of change is. And I think we saw this picture in last class. So this on the top is like the original sequence. It's here. We have what we call the silent point mutations, where the C here was changed by the T. So before the original sequence was TTC, now we have TTT. The RNA level now is AAA, but these two codons, AAG and AAA, code for the same amino acid. So there's not going to be any change at all in the protein. But what happens is if instead of changing the C, now we change the T by an A. So now we have T by A and then TC. Well, now when we transcribe that, we got UAG, which is a stop codon. So, and then we stop the production of the protein. And then we have what we call the missense mutations, conservative or non, where missense mutation means that we're going to change amino acid. The only difference is that the conservative amino acid change will have amino acids that have sequence very similar to the original one. If you see these, lateral change is very similar to this one, but in the non-conservative one, the new amino acid is very different. It may change the general structure of the protein, the charge. It may go from negative to positive amino acid, the interaction with other proteins. So it may change a lot of things. So depending on the type of mutation, it may be how it affects the protein synthesis or the protein production. And there are even more dramatic mutations that are called the changes in the DNA sequence, that are the chromosome translocations. So two chromosomes, different chromosomes, chromosome A and chromosome B, sometimes it can get very close to each other, and through sequences that are similar in between the two chromosomes, they can exchange parts. Though that's called translocation. And as you can imagine, if there is a gene, one part of the gene was in the dark chromosome, and the other part now it's in the other chromosome. So we can actually cut a gene in two parts, and now the gene is disrupted. And we're going to see examples of that later. So, and also something very important, I'm going to finish with this slide, I think, 2 more slides. It depends also, if you remember the structure of the gene, we have introns, exons, and we have the regulatory region of the gene. These regions, the regulatory regions, are very important because here, this is the region where many transcription factors, proteins, will bind here. The binding of those transcription factors will help to bring the RNA polymerase, in this case, the RNA polymerase, to this complex, and that will allow the cells to start transcription from here, to synthesize RNA. Okay, so if we mutate this region here, for example, the RNA polymerase is not going to be able to find the position where to start the transcription, and there's no transcription. So there's no production of the RNA, there's no production of the protein. If we mutate some other parts here, we may decrease or enhance the production of the protein. So where the mutation happens, how large the mutation is, what type of mutation is a change, is it a conservative, non-conservative, it's a silent mutation. So all those things are things that make molecular biology and genetic very complex, because it's not simply that a mutation will cause a disease or a mutation will cause a problem in the protein function. So it's just the same. And then the final one is, we were also saying that if the mutation happens in somatic cells or in germ cells, it will have even more dramatic effect, because germ cell mutations will be passed from generation to generation, whereas somatic mutations in somatic cells will just stay in that individual, and it may give rise to embryonic lethality if it happens during development, or predisposition to disease or death, or sometimes biological advantages to the organisms. Okay, so this kind of general introduction of DNA structure, mutations, and how mutations can be related to disease, it was just a very general view. So I will send you an email with the link that is this one here, and I can actually, just give me a second. I will see if I can copy and paste now for you that. Okay, I will paste it here in the chat, and I will send an email anyways. Okay, I'm just pasting that in the chat. It's a document where you will see some instructions. It's an exercise that you have to do to take a sequence. You will mutate that sequence and see how that sequence, that mutation may affect or not the structure of a protein. And I will give you instructions by email. And just to finish, I want to show you what's the what we're going to do next class. Okay. Just give me a second. So what we are going to do next class is, with that link that I sent you, I would like you to go and play around with that exercise. You are going to have some instructions. There's a link inside that document that will take you to a website where you can actually change the bases in a sequence, and you will see the effect of that in the protein sequence and function. So just go on that link, do that exercise, and then for next class on Monday, we're going to divide the session in two parts. One, we're going to discuss what you did. So what did you find with that exercise about mutating DNA? What kind of mutations did you find, etc. Okay, and that will be due in the first part of the Monday's class. And the second part, what I would like to do next class, I would like you guys to work in teams. We're going to make breakout rooms. Sorry, there's a mistake here, in romos instead of rooms. And I will send you a paper. It is paper here. It's not very recent, from 2007. And it's called A Silent Polymorphism in the MDR1 Gene Changes Substrate Specificity. In the journal is Science, and it's from 2007. So I will send you that by email. I would like you guys to individually read that paper. And in Monday, you will be separated in teams, and you will discuss about the paper. The discussion, you can do it in Spanish if you want. But then on Tuesday, one day after that, we will meet together in Zoom, and we're going to discuss all together about that paper. What did they do? What did they find? What was the techniques that they used? And it's an interesting paper because it's looking at the silent polymorphism that change is the substrate specificity of information by email. And I hope that probably by tomorrow, the platform will be, the virtual platform will be already on, and I will register you guys there, and then you will find all the information there, the PDF files for the paper, all this that I'm telling you is going to be there in the platform. Okay, so I think it's already 5 minutes past 8. So is there anything any questions or something you want to comment on today's class?
Doctor,
Yeah.
maybe for the Monday, you may